A pleasure speaking with all of you in this new mode of communication. What I want to do is show you ways that you can help your patients by identifying the causes of disease so that we now begin to understand how we modulate the infection, how we modify the biofilm, how we control the inflammation and the immune response. Now, we face a lot of unknowns. It's almost like the black screen that you see ahead of you right now. This has been something that's been facing humankind for a long time. About 400 or 500 years ago, we had an instance where, yes, they were wearing masks back then because they thought that the death was caused by a bad gas called a marasma, but actually it was the Black Plague. And this is what the Black Plague looked like. You died from it. Most people died from it. We had cholera epidemics. We have the Ebola. We have all sorts of different diseases that we could not control because we didn't understand what the causes were. Once we began to understand the cause, we could see the effects. And once you begin to do that, you can begin to understand the etiology of disease so that you can then customize your treatments so that you can now adjust with new education or keep doing what you've been doing in the past. And if it works, fine, but with periodontal disease, what we've been doing in the past didn't work that well. So let's take a look at exactly what has to happen for periodontal disease to occur. How do we get the shift from health to disease? Well, health is when you're in balance with the microbes that are in your mouth, whether you have symbiotic or commensal bacteria that are there. What happens, though, is the situations begin to change through a process called dysbiosis, and I'll explain that a little bit more in the next couple of slides. But you go from a balanced relationship to one that's out of balance, out of homeostasis, and that's when you get the microbial pathology that occurs. Some of the initial work on this was done by Philip Marsh, and in his article back in 1994, he begins to show us how oral health becomes disease. The first colonizers that appear in the human mouth are gram-positive aerobes. Then, as they progress, you begin to get secondary colonizers, and these are going to be more of your facultative anaerobes so that we can begin to understand this. What Philip showed us back 25, 26 years ago, this is what happens when disease occurs. The pH increases, gingival crevicular fluid increases, your redox potential decreases, and oxygen decreases. Now, some of you may not be familiar with what a redox reaction is. It's almost all the biochemical reactions that go on. Here's an example. If you take sodium and you take chloride, two independent molecules, and you put those two molecules together, you get sodium chloride or salt. When you put them together, the sodium gives up an electron, so it has a positive charge, and the chloride picks up the electron, negative charge. In humans, it doesn't work around salt, it works around hydrogen and oxygen. The hydrogen gives up the molecule, the oxygen accepts the molecule. And this is how the energy is derived, and this is what drives us. Well, as these four things occur, pH change, gingival crevicular fluid, your redox potential decreases, and your oxygen level decreases, what they found was your gram-negative obligate anaerobes become the predominant species. Looking at it on a more graphic aspect, the first thing that happens for periodontal disease to occur is that the pellicle, the proteins in the saliva, attach to the tooth surface. Then you get a thing called the Langmuir absorptive isotherm, which are just receptors from your gram-positive cocci, aerobic bacteria. They can attach to those. They use the sugar. They make lactic acid. We get an acidic environment, low pH. Then you get your secondary bacteria that come in, and these are where it begins to change. That's your secondary colonizers. most of these either gram-positive or gram-negative facultative anaerobes. In this environment, the pH is low because they're using the carbohydrates. They're making lactic acid. We get decay. This is what we learned. Well, as soon as the secondary colonists come on board, they begin to change this microenvironment. And now your predominant species become your gram-negative obligate anaerobes. So going back to what Philip was looking at, what we have to understand is the shift to disease occurs when these things happen. This is what we're going to discuss because I want to show you how and why the Periprotect method works. So here we have disease equals an elevated pH. Wait a minute, didn't we just say the pH was lowered because we had these bacteria using the carbohydrates and making acids and dissolving the tooth? Yeah, but we didn't continue to look at this. Look at what the research shows for periodontal disease. Instead of a low pH, in periodontal disease, the pH elevates, becomes 11.65. So you go from an acidic environment to a very basic environment. This also helps explain part of what Lou was talking about with the gingival caries. You don't have caries down in the periodontal pocket because you got a negative pH. You have such an elevated pH. The bacteria that cause decay can't go down there and make enough acid to erode the root surface, which is why when he's talking about using MI paste, he's talking about using fluoride gels. Makes perfectly great sense that you can find a way to control this down the road. Well, let me show you, if the pH is elevated, 11.65, what does hydrogen peroxide do? Hydrogen peroxide actually lowers the pH. It reverses the elevated pH to a lowered pH. Well, what about gingival crevicular fluid? Research shows that hydrogen peroxide lowers the gingival crevicular fluid. In essence, it reverses the process of disease to a lowered gingival crevicular fluid. All right, the redox potential. Well, it's decreased in disease, but look what hydrogen peroxide does. It's one of the most powerful oxidants. It reverses this disease process. It elevates the redox potential and reverses the process of disease. And obviously, hydrogen peroxide breaks down to water and oxygen, so you can reverse your oxygen decrease to elevate your oxygen presence. When you reversed what happened in disease, you lower the pH, you lower gingival crevicular fluid, you increase your redox potential, and you increase your oxygen, you go from disease to health. So what we're showing you is just with the addition of a simple compound such as hydrogen peroxide, you can reverse this whole concept or process and reverse the microenvironmental changes that had to occur for disease to occur, reverse that so health is going to be an inevitability. Now let's talk a little bit about the bacteria and the changes that they make with this as well. Now, how do you determine what microbes are present? Well, you can culture them. This is what I learned, I hate to admit how many years ago, when we first started looking into bacteriology and microbiology. The problem is you cannot culture very many of the oral bacteria. You can only grow about 40% of them. As soon as you take the anaerobes out, you put them on a petri dish, they're not going to grow anymore. So this is a highly inaccurate. You have greater than a 50% error ratio of trying to find out what's there. Well, around the 1970s, 1980s, they came up with a concept of a fluorescent in situ hybridization method, which is called a PCR, using probes. Now, these were great, but they required a separate probe for each bacteria that you're looking for. So if you're looking for PG, you had to have a PG probe. Looking for AA, you need an AA probe. FN had to have an FN probe. But that's what they had in the beginning, and that's how they came up with this list of bacteria responsible for disease. I'm going to show you a periodontal pocket that doesn't have one of these in it. Are these a cause of disease? Well, maybe, but let's look at how this was derived. How did they determine these bacteria? These are the 4 articles that are responsible for how we came up with these 11, now it's up to 16 bacteria. Ladies and gentlemen, these were published over 25 years ago. If I had a physician come into me and say, okay, you have a carbuncle on the side of your face, and I'm going to use data from 25 years ago to tell you what's there so we can treat it, I'm going to let my feet do the talking, and I'm going to walk out his office, and I'm going to go to some other office. I don't want a test that's 25 years old. You see, polymerase chain reaction tests only will tell you what they're looking for. They don't tell you what's there. And I got news for you. Periodontal disease is a heck of a lot more than the bacteria that are 11, 12, or 16. When we started doing the work with microgen sequencing, oh my gosh, this next generation sequencing, it just opens an entirely new avenue of recognition. Now, you do have choices. You can do your traditional culturing methods. There's nothing wrong with that, but you're only going to get about 40% of the bacteria that are there. PCR will only tell you the ones they're looking for. Our next generation sequencing will really open the door for you to understand what you're looking at. So why do I recommend the PerioDX system? Well, first off, it is the most accurate test out there, bar none. It will tell you up to 30,000 possible sequential possibilities of the different microbes, virus, fungus that are there. It doesn't lead us down the pathway that some of the diagnostic methods do about saying you need this antibiotic, that antibiotic. A couple of years ago, the CDC came up with a guide that they are requesting healthcare providers do not prescribe antibiotics unless it is in emergency instances. And it also works with your prescription trays. I'm going to show you some customized treatments we never would have been able to do if we were not using this PerioDX system. It is a phenomenal system. Now, it uses 2 concepts. It uses a salivary rinse and a pocket sampling. Understand, the saliva samples are just not representative of the bacteria that are in the pockets. PCR determines the concentration of microbes. Next generation sequencing tells you what's present. So when you do that, what you're going to do is, and I want to show you a way that we have found the best way to get your saliva sample and your pocket sample as thorough as possible. When we get the kit from next generation, from Microgen, for the PerioDX, we take the solution that is there, we put it into a syringe, and we lavage out all of the deep periodontal pockets in telling the patient not to swallow it. Then we have the patient swish their mouth with this solution, expectorate it into the collecting apparatus, and we ship it to Microgen for analysis. So we've got the saliva, or we've got the solution, we flushed out the pockets, we know what's in the mouth now, and then we go back and we individually, for a pocket or all of the deeper pockets, we basically suction out the bacteria that are there so that we can begin to get a sample from the patient themselves so that we know what's there. So what you do is you elevate the plunger for 10 seconds, then you remove the micropipette from that periodontal pocket, And if you're just going to evaluate just the one pocket, you unscrew the little tip, put it into the shipping container. And if you're going to evaluate 2, 3, or 4, you would just use the same tip to go into 2, 3, or 4 areas so that you can evaluate what's there. You send it to Microgen, then they send back to you the printout from the analysis. You'll get both a PCR analysis and your NGS analysis. With this, you're going to know all the microbes that are present. You're going to be able to target now the source of infection, not just the 11 bacteria somebody might think are there, because now we can identify them all. Now, PCR in this case did not correspond with next generation sequencing because they did not do the lavage technique for this patient. They had the patient rinse with the material and they got what was in the saliva. Well, it didn't even come close to finding out what bacteria were in the next generation sequencing. So I think salivary analysis, you have to be very, very careful. It's not going to tell you what bacteria are down in the periodontal pockets. When you get the results back, though, boy, do you get a hodgepodge. I happen to be sitting at my desk, and I have the original paper in front of me where I began to correlate and make a record of all the different bacteria. And I think we had like 300 of them that we started to find in these periodontal pockets. And it did not make any sense. So what I did was I started to look at them, and this is probably a 3-month period of time. I began to see that I found the gram-positive bacteria in more healthy areas. I began to find the gram-negative bacteria in more diseased areas. So I began to divide these hodgepodge of bacteria into gram-positive and gram-negative. Then what I did was I took a look at what categories. We had anaerobic bacteria, facultative anaerobes, aerobic, and fungi. And when we did this, all of a sudden it started to make sense. And we published this for you in an article that Tanya has made available for you. It's the composition of microbes in periodontal pockets. I work with Dr. Bruce Cochran from Iowa, and Bruce is just a superb periodontist. Actually, I think he's a prosthodontic periodontist. He's one of those odd ducks that like to go to school a lot. What we found in his patients, healthy patients had certain characteristics. Gram-positive bacteria, now this is a 3 millimeter healthy patient. They didn't have any pockets in the mouth beyond 3 millimeters, but this is what we found in this 3 millimeter pocket. Gram-positive bacteria were greater than gram-negative, and facultative anaerobes and aerobic bacteria were greater than anaerobes. What occurs in disease pockets, 3 millimeter pockets in disease patients, and this goes back to what Lou was talking about, was if the bacteria there around one area, they're able to be around all the areas, including your implants. But what happens in disease, the gram-positive bacteria decrease, but the gram-negative bacteria increase. Your anaerobes increase as your facultative anaerobes and your aerobic bacteria decrease. And we found this over and over and over again. So what you found was that gram-positive decreases the deeper the pocket, the more the gram-positive decreases. So you get a decrease in your gram-positive bacteria. There is also a decrease in your facultative anaerobes and your aerobic bacteria. There is an increase in your gram-negative bacteria and a tremendous increase in your anaerobes as well. This is the bacterial change from health to disease. So here's what you're looking at when you're looking at a patient that has a healthy situation as compared to a disease situation. Now, do you want to look at all the individual members that are here? Probably not. So let me show you what next generation sequencing, PerioDX, Microgen DX has done. They've put this graph on the chart for you. You don't have to look up all these bacteria and figure out what they are. It's here in a graphic form. You can see there are no gram-positive, well, 6%. There's 73% gram-negative. There's very few aerobes, facultative aerobes, and your anaerobes are the predominant species. Glancing at this chart, you can go, okay, I understand what's there. Now, why are gram-negative obligate anaerobes found in most of your pathological situations or conditions? What is it about a gram-negative bacteria that makes them so virulent? Well, let's look at the difference between gram-positive and gram-negative bacteria. Gram-positive bacteria have a dual cell wall, cell membrane. Gram-negative have a trilayer. They have an inner membrane, they have an outer membrane, and they have the periplastic space between them. With this makeup, what gram-negative bacteria can make is to make lipopolysaccharides on the outer membrane. And these lipopolysaccharides are what make the gram-negative bacteria so virulent. Now, if you have a gram-negative bacteria and it gets into your bloodstream or one of your white blood cells, ingests it, it can kill the bacteria. It can dissolve the membranes. It can dissolve the periplastic membrane. It'll take care of all the lipids, carbohydrates, proteins, etc., but we don't dissolve the lipopolysaccharides. So they get released into the environment. Body doesn't like that, so you have protein binding materials, such as your interferons, which can be recognized by any of your autoimmune cells, such as the macrophage. Through a toll-4 gate, that lipopolysaccharide is introduced into the macrophage, then through a process called transcription if it's RNA, transduction if it's DNA. It travels to the endoplasmic reticulum where it only makes pro-inflammatory chemokines and cytokines. It only starts inflammation.